2723, this one right here?
Yeah. Because I just like seeing them similar to me.
Oh, compare the two. Let's see, 27, it's 28. All right. This one is about, this is a schematic showing how, showing the steps that have to happen if a virus has a positive RNA genome. So the positive RNA genome is represented in green. And it's showing that the genome, after it enters the cell, serves two purposes. One purpose is for use as a template for translation. That's right here, and this red squiggly line represents the viral proteins. And the other thing that it has to do is serve as a template for making negative single-stranded RNA. That's what this line is. And then the negative single-stranded RNA has to serve as a template for making more green positive strand. And I wrote in a whole bunch of these. This crap here is my writing with a green pen. And I put a whole bunch of those in there to show you that it's not as if you just think of this, this is not meant to be stoichiometrically accurate, right? There has to be a whole bunch of these in order for there to be reproduction of the virus, because there's going to be a whole bunch of virions made, and they're all going to have positive RNA in there. And in the meantime, some of this positive RNA is going to be used for protein. So there's positive RNA, or mRNA, on both sides of this glob here, showing that the template that comes in is used for making protein. The genome that comes in is used for making protein, and also this transcript that gets made is for making protein. Remember what we said about different classes of proteins or different categories of proteins that viruses have? It has to do with time. What are you thinking, Luke?
Well, like the immediate, early, late proteins.
Yep.
Used to for the state of the cell cycle, I guess.
That's right. So the immediate and the early and the late. So this arrow here represents an immediate protein, because this is something that has to be made immediately as soon as the virus gets into the cell. When it goes this direction, they're referring to late proteins, because this is coming from a transcript that got made after the virus was in the cell. So when the proteins that get made in this case are probably the capsid proteins. And then this arrow, this is kind of a messy figure. This arrow, this short one, then shows that the virion is getting packaged with this as its genome. This long arrow represents the protein being folded up and being used to make the capsid. Tony?
So what do you say the arrow from the plus RNA genome on the right represents?
This one?
Yeah.
This represents the translation of the late proteins.
Okay.
So, because this protein has arrows coming at it from both sides.
But the one on the right side, the positive RNA genome, is transcribed from translated or transcribed from minus RNA, right?
Yep, that's what's going on here.
So do positive RNA genomes that are transcribed from the minus RNA that only make late proteins?
Do positive, well, you would say that it would be true the other way around, that the late proteins are made from the transcript coming from the negative strand. But it could be that there's some intermediate ones. Because remember we had the immediate early, the early, and the late. Whoops, this is in a DNA virus. Let me find an RNA virus. Let's see here. They don't differentiate in here for this DNA virus, or for this RNA virus, I mean. So it would be safe to say that, where'd that go? What figure were we looking at here?
728.
There we go. It would be safe to say that this represents late proteins for sure. Whether that in between stage fits in, they don't really tell us with this. See what I'm saying? So if you know that if you have a late protein, that's definitely going to be.
So is that left? Is that RNA genome on the left not going to make late?
Right. That's not going to make the late proteins. The late proteins aren't going to get made until it's got the genome all replicated and it's ready to package it up.
Another question.
the enzyme might be the same. In some cases, one enzyme does both of those activities, but the function is different. So I don't really appreciate the way the book describes it, because it kind of, here's where it is. RNA-dependent RNA polymerase. The cells have an RNA-dependent RNA polymerase, which uses an RNA template to make a new RNA molecule. When the enzyme is used to replicate the genome, it's called the replicase. When it's used to synthesize the mRNA, it's the transcriptase. So they are, rather than calling the activities two different things, they're calling the enzyme two different things, even though it's still the same protein, which I think it's helpful in terms of learning the functions of the protein, but it's not very helpful because then people feel like they have to label the protein different things. Go ahead.
And the book says when it replicates a genome, it's called replicase.
Yep.
But it also needs to do that. It's making the negative strain RNA, the replicases. But that
Yeah.
won't be the genome, right?
Well, it has to do that in order to get to the
Yeah.
genome. So this would be part of the function of making the genome. Where it really gets annoying is this. This arrow, why is it so crooked? It's making me nauseated here. This arrow represents replicase and transcriptase activity. So the whole thing just gets to be a little unfortunate. But as long as you understand the purpose of the enzyme, then it doesn't really make any difference, right? So what was the second part of your question? This, let's see here. Was it 28, 27, 28? So in this case, this is a situation where you have a negative RNA genome. These two arrows represent the activity of RNA-dependent RNA polymerase making a positive strand that's going to be used as mRNA and a positive strand that's going to be used for making more negative strand. And again, I drew in a whole bunch of extra red things here because that is more representative of the fact it's going to make a whole bunch of different copies of its genome, right? So in this case, they drew the green line twice, showing that they have two different functions, but they really are the same molecule. Christian?
Is there an evolutionary advantage to storing the coding strand, which is the template strand?
Is there an evolutionary advantage of doing what?
It's like storing the positive strand at your genome versus the negative strand. Because it seems like a stages step would be quicker.
I don't know if there's an evolutionary advantage, but I would think there would definitely be an advantage in terms of likelihood of, well, actually, you have to, the problem is, I would think that there would be an advantage to having a double-stranded genome. I'm not sure if you could pick whether positive strand or negative strand is advantageous because you have to make the other one anyway. And if you have an error-prone enzyme, that's going to get screwed up. I would think the advantage would come in when you're double-stranded because then you would have to have some level of fidelity in order to make it work at all, right? Because they have to match up. And then the incoming virion has both templates, the top and the bottom. So
I'm not sure what the advantage of being positive or negative would be. That's a good question. Anybody else? Let's see here. Other questions about chapter 27 or chapter 6? Okay, we're going to go back to chapter 3. Last time we started talking about cell biology of prokaryotes. And we talked about cell shape and how the descriptors, the vocabulary used for cell shape also is used for naming the cells. Sometimes, what that happened? Here it is. Sometimes the name of the cell is based on the shape. So let's talk a little bit about naming before we get too carried away here. When you name an organism, of course, you have a genus and species name. Here's an example, Bacillus. I'll write in red so we know it's a new day here. Bacillus and rhesus. And, whoops, I spelled that wrong. Right. And rhesus. Notice that the genus is capitalized, the species is lowercase, and the whole thing is underlined. This is an example of an organism that's genus is named after the shape of the cell, right? So you can guess that this was named a long, long time ago based on the fact that it was the bacillus, or this rod-shaped cell, that caused anthrax. So sometimes the organism is named after the disease it causes. Sometimes it's named after the shape of the cell. Sometimes they're named after people, which is a little bit annoying. Sometimes they're named after their characteristics. Here's a really good example of one that's named after its characteristic. Let me write on the back. Use your notes. It's Thiomargarita namibiensis. Notice I'm underlining the whole thing. You guys need to remember to learn to underline this stuff. You want to be at least as good as USA Today. You know, the newspapers always screw this up, and they write E. coli, or they write E. capital coli. It's a terrible mess. You don't want to be those guys, right? Thiomargarita namibiensis. Thio, what is thio? Sulfur. What's margarita? It's not that. Margarita means pearl. Thiomargarita namibiensis. Namibiensis refers to the country of Namibia. It's a giant microbe. It's so big you can see it with your naked eye, that has sulfur granules in it. And it was named margarita because the sulfur granules made it look all pearly. And it was found off the coast of Namibia. And it's relatively recently discovered, just in the last 15 or 20 years or so. So if you discover an organism, you can give it a really nice, handy descriptive name. You can name it after a person. Whatever you do, write it with a capital genus and a lowercase species and underline the whole entire thing, right? That way you don't drive us nuts. So we're going to talk about some characteristics of bacterial cells today. There's going to be a reading assignment for you guys about size. Did you guys all see I put a reading assignment on Moodle about cancer? It's on there. There's going to be one about size. Microbes have to be small. And it has to do with the surface to volume area. Read about that. We're going to talk about some common parts of bacterial cells. We're going to leave out the stuff that eukaryotic organisms have. So one thing we're not going to take the time to talk about is ribosomes. We're not going to talk about it now for sure. We're going to talk about ribosomes later as they pertain to taxonomy and phylogeny, but we're not going to talk about the function because you've learned that a few times before. So we're going to talk, here's a cartoon. You should be able to diagram. This is kind of a boring figure. It doesn't have a lot of detail in there, but you'll know all the parts of that when we get done here. We're going to start out talking about the plasma membrane as it pertains to bacterial cells. Square this baby up. Okay, when you're learning new stuff, it's always a good idea to assess what you already know, right? What do you guys already know about plasma membranes from cell biology? Ben?
Your lipid bilayers?
The lipid bilayers, yep. The phospholipid bilayer is the main part of it, or it's a major component. How about the function?
Semi-permeable?
Yep. It's a semi-permeable barrier. Just as it is in eukaryotic cells, it's a semi-permeable barrier. One of the interesting things about it, and from the point of view of microbes, of bacteria, the plasma membrane is where it's the border between the inside of the cell and the whole environment. With our cells, in a multicellular organism, most of the time the plasma membrane is the barrier between one cell and the next, right? It's not really the whole entire environment. And so there's lots of functions that have to happen in the plasma membrane besides just separating the inside from the outside. And those functions are catalyzed by the proteins that are inside the plasma membrane. So like Ben said, the phospholipid bilayer major component. What do you remember about that? The phospholipid bilayer. What's the word that describes phospholipids in terms of their chemical nature?
It has a phosphate group on
It
it.
has a phosphate on it, which is charged and consequently polar. And then what about the rest of it?
Hydrophobic tail.
It's hydrophobic. So one is hydrophobic and one is hydrophilic. And what's the vocabulary word for that? Yeah. I heard you whisper that. I'm having a hard time spelling it right off the top of my head. Amphipathic. Meaning that it's got, both in one molecule, it's got a positive polar side and a non-polar hydrophobic side. And that's what's shown in here. You guys have probably seen this 1,000 times before, starting when you were in grade school, probably. Let's look at a little bit more detail on here. Just because I want you guys to get a feeling for, like, how would you diagram this in a test? Theoretically, if you were asked to diagram the cell wall and the plasma membrane, you don't have to draw all of these, but this portion up here, this round part, is the polar part, the hydrophilic part. It's got ethanolamine in there, it's got a phosphate group, it's got glycerol. All of that stuff is polar and hydrophobic. It's even charged. Then it's got these long nonpolar chains, and in this case, they're shown all totally saturated. There can be a whole bunch of different levels of unsaturation. There can be differences in length that the microbes do if they need to alter the permeability of the membrane because of high temperatures. You guys are going to read some stuff about what the archaea do in terms of their membranes, because remember, they live in really extreme temperatures. They've got a bunch of modifications to tolerate the fact they're living in water that's hot enough to boil. So if you're going to draw this, you draw it something like this, right? You would represent some bonds in here. It's got a head. This little shoulder thing represents the glycerol, right? And you would, if you were to draw the bilayer, of course, draw them like this. So why are these associated with each other? So some of you guys are thinking, oh my God, we've heard this 1,000 times, but I've taught this 1,000 times. And I know that some of you need to hear it again. Why are these associated with each other? What, Tony? Not only the other end, but also by the water itself, right? By the aqueous environment. So when you draw, if I say draw a schematic diagram of the plasma membrane, you wouldn't do this, would you? I hate to even draw. Don't put this in your notes. Do not write this. I've seen that before, you know, over and over and over. Here's my favorite. And I'm not saying this to make fun of the students that did it. I'm saying this to make sure you know this is not correct, right? Because we're all learning. Those would not be correct. You can't have a membrane made up of that. It's got to be this way. And when you are under pressure someday, you're going to get screwed up. Okay. So we'll talk about variations in permeability and stuff when we talk about some different things in terms of tolerance of heat, when we talk about different kinds of environmental things. Okay, what else is out there? The really important stuff in terms of functions are the membrane proteins. So think about all of the functions that have to go on in the plasma membrane of single-celled organisms, of bacteria. So what kinds of things do they have to assume? They have to do all the things that our plasma membranes do, which includes transport. They're responsible for getting everything across. Proteins for transport. So there's a whole bunch of stuff in the book about transport, and I know you guys did cell biology, right? The symport, and the antiport, and the ABC transporters. We're not going to go into all that stuff again. Just know that bacteria have all the same types of transporters, except they can't do endocytosis. So they can't take things in by great big huge gulps at a time. They have to take things in one stinking molecule at a time. So if they need to take up peptides, they have to have a transporter that brings the peptides in. And there's limits to how big those things can be. Same thing goes with sugars and ions, and anything they need to take in, they have to take it in by small parts. What other kinds of functions would have to happen in the plasma membrane of prokaryotes? Motility? What? Motility? Motility, actually, yes. Motility would be involved in, would have to do with the plasma membrane and also with the cell wall. And in fact, it's not just motility, but the ability to detect the things it wants to swim towards, right? So let's write motility and chemotaxis. Especially the sensors. So when bacteria swim, they don't just swim randomly, they swim with a purpose. And they have proteins in their plasma membrane that are used to detect the stuff that they want to swim towards. We'll talk about how that works. It's really amazing. Talk about that later. What were you going to say, Tony? Yeah, that's what I was hoping somebody would say. Electron transport for respiration. Also, you might not have thought of it, but also for photosynthesis. Lots of prokaryotes are phototrophs. You'll see that in your Winogrosky columns. All the different colors are from bacteria that are phototrophic, and they have to do all of their photosynthesis in the plasma membrane because they don't have chloroplasts, right? In fact, for both of these two, the bacteria are taking the place of, or they're taking on the roles that happen in our mitochondria and in our chloroplasts, right? Did you guys learn about the endosymbiotic hypothesis in cell biology? What was the deal on that? Endosymbiotic hypothesis. Right. Exactly. Eventually, so what he said was that microbes got swallowed up by some early eukaryotic organism, and the functions of respiration and photosynthesis actually came from those bacteria that got eaten. And so, of course, all those functions have to still be present in the bacteria, and in that case, it's present in their plasma membrane. So in some cases, okay, so functions about the plasma membrane, that's stuff you've heard about before, nothing too exciting. Some prokaryotic organisms, let's see here, let's label this, we label this one last time. Cell shape, this is 2, plasma membrane, 3, internal membrane systems. So one of the things we like to call defining about prokaryotes are their lack of internal membranes, which is a little bit ironic because some of them really do have internal membrane systems. Here's a picture of a couple of organisms that have internal membranes. Let's see here. So there are fewer in prokaryotes to be sure, but the internal membrane systems are extensions of the plasma membrane. There's just lots and lots of extra folds, and they're especially present in organisms that have to do the functions that we were just talking about, photosynthesis and electron transport. So sometimes they are sort of thylakoid-like, taking the place of the thylakoids, which gives them a site for photosynthetic machinery. Or it could be a site for electron transport for respiration. This is especially true with anaerobes that are doing respiration of nitrate, using nitrate as a terminal electron acceptor. They need a whole bunch of more space. We're going to talk more about that when we talk about different kinds of respiration besides aerobic respiration. Now let's see here. I think what we're going to do, I'm going to put this in your notes because you're going to do this yourself, but I want to make sure you do it. You're going to get a reading assignment that has to do with this category, and those are the inclusions. Sometimes they're called inclusions, sometimes they're called inclusion bodies. So I wanted to say just a little bit about it to avoid some confusion. When inclusions were named, it was based on the fact that when you look at bacteria under the microscope, lots of them have, if you look at them, you see chunks in them. And people didn't know what the chunks were. They just knew there was something inside of there. And they called them inclusions because anytime you see that an object has some thing stuck inside of it, you can call it an inclusion. Like if you look at a diamond under a microscope and you see some chunk of coal or some fleck of something in there, that's called an inclusion in a diamond. And people didn't really know what they were in bacteria. It turns out there are a few different kinds of inclusions that can possibly be there. There are some that are used for storage, some that are used for buoyancy. There are some that are used to orient the organism with respect to the Earth's magnetic field. Those are called magnetosomes. And there's a video on Moodle that you can watch about the magnetosomes. You guys are going to do some reading about that, but I wanted to make sure that you understand that it's not just anything that's a chunk inside the cytoplasm. They're ones that are big enough to see. So every year somebody puts down that ribosomes are inclusions, and they're not inclusions. They're ribosomes. They're a structure. And you can't see them under a light microscope anyway, so I'm not sure how you would think ribosomes are inclusions. I'm going to put not ribosomes here for good measure. Okay, so I want to talk about the most important part, according to many textbooks, the most important part of cell biology of microorganisms, many books say, is the cell wall. And it is super important. It's hard to say it's the most important part because there's some weird microbes that don't have any cell wall whatsoever, but it's important to us for a lot of reasons, and it's important to the microbes especially because it gives the cell strength against osmotic forces. So it's easy to forget, because of the way we grow microbes in the lab, it's easy to forget that most of the time the environment outside of a cell is hypotonic relative to the inside of the cell. Because the inside of the cell, when you think about the inside of any kind of a cell, you guys typically picture it as, let's see the picture, let's see what they, look at this picture. They show little chunks of things in there, but they give you the impression there's a lot of loose space in there, but actually it's super packed, it's viscous. If you could stir it, if it were big enough to stir, it would be thick. And so it's really, really concentrated with protein and potentially sugars. And so the cell wall is actually used to protect the cell from osmotic forces, because what would happen, typically, if water could get across there, it would cause the cell to expand and lyse open, right? Because the water would have a tendency to go towards the higher concentration. So the cell wall is responsible for protecting against osmotic stress, for one thing. It's, in terms of human interest, it's medically important for a few different reasons. One of the main things you think about in terms of the medical importance of the cell wall is it's the site of action for many antibiotics. One of the interesting things about that, though, is that it's not that the antibiotics attack the cell wall and tear it apart so much as it is that they prevent it from being built. So we're going to talk about how that works and examples of antibiotics and how they're involved in preventing the synthesis of the cell wall when we talk about anabolism. So we're going to just talk about a few things about anabolism, and one of them is the cell wall. So we'll talk about how they work. But for now, just know that if the cell is trying to grow in the presence of antibiotics, the cell wall is going to be weak, and the whole thing's going to be susceptible to lysis. It's also important for pathogenicity. Pathogenicity, I don't think we defined that. That is the start of disease. It has the word genesis in it right here. Pathos refers to disease. It's important for establishing disease. And there's a few reasons for that. One of them is that it has, in some cases, it has some proteins that are involved in adherence. At least it would have them attached to it. Another part of it is that it has a virulence factor inside of it, we're going to talk about in a little bit, which is LPS. I'm not going to say much about it now because we're going to go into detail later. It's a virulence factor. It's actually part of the bacterial cell wall. in terms of classification, it's also important because it's the basis for early classification. That is Graham, the Graham reaction. So a guy named Christian Graham figured out that, so imagine if you had really early microscopes and you didn't have phase contrast microscopes, so you had to stain everything in order to see it. One of the really important stains that you guys are going to learn to do in the lab is called the Graham stain. And it's important because it allows you to differentiate between different kinds of bacteria based on the interactions of the stains with components of their cell wall. So if you have something that's Graham positive versus Graham negative, that tells you something about the way the cell wall is built. And as it turns out, people are starting to think that maybe Gram positive and Gram negative aren't really the best descriptors, and there's a section in your book that talks about how they now think maybe they should call the bacteria monoderms and diderms based on whether they have one layer or two layers, because it's not, taxonomy is not totally neat and tidy with respect to the classification. So we're going to look at the difference here between the classification. The Gram-positive ones, you guys, some of you guys have done this before. Gram-positive stains blue, or do we call it purple? Purple? I've been told I can't do colors. I've been told. My wife says I don't know the difference between different colors. Gram-negative, I know that stains red. And here's why. This shows a diagram of gram-positive cells and gram-negative cells. So in the gram-positive cell, you have the plasma membrane, which is shown here in red, tiny little thin strip. This stuff over here is the cytoplasm, and it has this orange staining area, which is the cell wall. It's composed of peptidoglycan, which we'll talk about in detail here in a second. Gram-negative bacteria have a plasma membrane shown here in red. It's got the peptidoglycan, which is a very thin little layer shown right here, and then it's got on the outside of that another membrane right here, plasma membrane out here. So when you use the Gram stain, it stains the Gram positive, positive for staining, blue, because of the fact, or purple, because of the fact that the purple stain gets trapped in this big thick layer of peptidoglycan. So you treat the cells with the purple stain, you add iodine, which complexes with it, makes it insoluble, and it gets trapped within this network out here. And we'll look at that network in a second. With the Gram-negative cells, you don't have that big, thick area for the blue-purple stain to get trapped. And so you can detect them with a counter stain, which is red. And that's all based on not anything more than just the characteristics of the cell walls of the bacteria. So there's two major categories. So we're going to look at some detail about what's in the cell wall and what makes them different with respect to their staining. And the main thing, like I said before, is a molecule called peptidoglycan. So you can probably guess, based on the name, you can probably guess what it's made out of. It's a complex polymer. It gives stability. The cool thing about it is that it's relatively porous. Porous. Would that be OUS? So things can get through there. It's sort of, I like to think of it as being sort of like chainmail. You know, it's strong and it's rigid, but it's got to be flexible at the same time, and stuff has got to be able to get through there. It's composed of two different types of molecules, as you can tell by the name. It has sugar associated with it. And it also has amino acids. So the two sugars, let's look at the sugars here. Here's a structure of peptidoglycan. This is one monomer of peptidoglycan. And the sugars up here at the top, you guys recognize these as sugars. You don't need to memorize, although you will, some of you will be able to memorize the names of them, but you can memorize the abbreviations. NAG stands for N-acetylglucosamine. This is glucosamine. There's the amino. There's an acetate up there. And this one is N-acetylmuramic acid. So where's the muramic acid down here, right? Or N-acetyl. Here's N-acetyl right there. Here's the acidic part. So there's a chain of these, a repeating chain. A subunit has two of them that are linked together right here. And then hanging off of the N-acetylmuramic acid is a chain of amino acids. So some of these amino acids are weird. Let's write down a couple of things here before we get carried away. NAM and NAG are the two sugars. The amino acids have some different properties. They're not amino acids that are used for translation. Is the major difference. Some of them are unusual. One of the unusual parts about it is that some of them are D amino acids versus L, right? They're not L amino acids. So what's the deal with D and L? Do you guys remember that from organic chemistry, or where would you get D and L?
The light, it like how it rotates light.
It has to do with how it rotates light, and in terms of functionality, they're non-superimposable mirror images, right? They're stereoisomers, and the significance of that is that the L amino acids are the ones that are translated into proteins, and the bacteria put D amino acids into their peptidoglycan, which makes them resistant to the standard proteases that exist in the world that would chop up proteins. So these are little peptides that are made not on ribosomes. They're not a product of translation. They're assembled by enzymes. And the fact that they're D amino acids means that they're resistant to proteases that would break down a typical protein. So here's D-glutamic acid right here, D-alanine. It says at this site, we'll talk about that in a little bit. Here's another D-alanine. This is an amino acid. It's a weird amino acid. It's diaminocamelic acid. So it's got a carboxyl group and an amino group here, but then it's got this bizarre thing out here that's not even something you would put into a protein. It's still amino acid based on the definition, which means that it's not going to be something that gets recognized either in protein. Okay, so this is one subunit of peptidoglycan. And what happens is there's another chain of these here. There'd be another NAM attached with an NAG and another peptide chain. Down here at this site, at this terminal D-alanine, it says at this site a bond may be formed to link this peptide to one on another chain. And when that happens, this gets taken off. Let's look at that while we're talking about it. The result is something like this. So you have opposing chains of peptidoglycan that are linked to each other. There's a whole bunch of different ways, and I think I might have thrown this figure out because we're not going to hear how I did throw it out. There's a whole bunch of different ways that different organisms do it. So they can either have a direct linkage, which is shown here, or they can have an interbridge with 5 amino acids or a different number of amino acids between the two. And this figure shows a bunch of variety that exists in the world. You don't need to learn the variety. The point is just that different organisms have different ways of figuring that out. So the polymer forms, it assumes this great big huge helical thing, and then there's many, many helices of these all attached together. The blue circles represent the peptide side chain, and then they get cross-linked covalently to other peptide side chains, and you get a gigantic polymer that forms around the cell, and it's so strong that you can even lyse it really carefully and get the guts of the cell out and still have this sac of peptidoglycan left behind. That's what's shown in this figure. Let's see here. So this is a picture of all those things being associated together in the gram-positive cell wall. These represent the sugars, the orange and the yellow. These represent the cross-links of amino acids. So the amino acids are essentially not shown to scale. But the idea is it's a great big huge thick pile of peptidoglycan. And the purple stain you use in lab, the crystal violet, gets in there and complexes and becomes insoluble and can't be washed out because it gets trapped inside of here. And here is the association of it with the plasma membrane. So one of the components of the cell wall and gram-positive that's important for keeping it attached to the plasma membrane is what's called lipoteichoic acid. It links the peptidoglycan, I'm abbreviating that PG, to the plasma membrane. there's something I want to forgetting to tell you about the plasma membrane. So how would you draw peptidoglycan? Let's go back to this. I know what it is. So for the level of detail of drawing peptidoglycan, you could do it at this level of detail. You wouldn't have to draw it helical, but you would be able to draw something like this. NAM. If this were a biochemistry class, this would not be acceptable, right? Let's go back to where it's not curly. That's screwing me up. NAM. Something like this, right? You have repeating subunits of it. And then the NAM has amino acids coming off of it, like this, right? And they would be cross-linked to other NAMs, either adjacent or next on a opposing NAM, like this, right? Because you're going to have a huge pile of it. So there would be a linkage between here. And it could either be a direct covalent linkage, or there could be a whole bunch of amino acids in there, right? Depends on what organism you're talking about. So this whole thing gets constructed, gets put together. There's several places within this synthesis that antibiotics can prevent the activity. We'll talk about that another time. Let's see here. Any questions about the basic structure of this? Here it is with the gram-positives again. Shows the plasma membrane down below, right here. This little tiny space in here is called the periplasmic space. And in gram-positive bacteria, it's relatively small. What you can't see in here, what they don't show, is that area, and also linked within the cell wall, the peptidoglycan, are a whole bunch of enzymes. Enzymes involved in putting the cell wall together, enzymes involved in chopping up polymers that the cell wants to take up. So bear in mind, remember how we said that these cells can't do endocytosis and they have to bring things in a molecule at a time? Imagine if these are transporters and it's bringing molecules in, there has to be some kind of a hydrolase out here that chops them up into small enough pieces, like proteases or maybe cellulases or amylases, chopping up the things into monomers that can be brought into the cell. Not shown in the picture, but super important. So we're going to do Alex's presentation. Does anybody have any questions on your mind before we do that? We'll do, I will probably start talking about gram negatives next time. Let's see. Sure, here we are. Did you think I forgot?
A little bit, maybe. So the microbe that I chose to study is Photorhabdus luminescens. It is a bacteria that lives inside soil nematodes. So it is swallowed by the nematode, lives in the nematode, hangs out. It naturally fluoresces green. So this picture shows what a nematode looks like when it is, I don't know if infected is the word, but when it contains the luminescence bacteria, it glows green. So during the Civil War, they called it an angel glow because they saw a lot of it in the soil there, and then people would turn green because it would be in people. However, it produces antibiotics, naturally produces antibiotics, and those antibiotics keep wounds cleaned, apparently. They're not exactly sure, but it doesn't infect or make humans sick or anything. So they would use that for wound packing in the Civil War, so then people would turn green because it naturally fluoresces green. So it's super, super rare for it to cause disease. Occasionally it does, just like everything does if someone has a weird reaction to it. But for the most part, it's harmless to humans. So it lives in the nematode after it is born, and then the nematode will infect an insect, so it'll worm its way into an insect, and then it'll spit the photorhabdus luminescens out, and the bacteria will release all of its toxins, and then after it releases the toxins, the insect dies, and they rapidly reproduce using the organic material inside the insect, and they like eat their way out from the inside, like bursting through. So it's like kind of like an alien movie where the alien pops out of the human, very much like that. So after it pops out, then it just does the same thing. It finds a nematode to hook up with. The nematode infects an insect, and we do the whole thing over again. So very peculiar, but it works for them. So my take-home points are just that it lives inside the nematode, and the nematode is what infects the insect. However, the nematode and bacteria both reproduce using the organic material from the insect, and then they reproduce until the insect blows up.
Did you say that they packed the wounds of Civil War?
They would kind of not pack, but like they would like use, yeah, they would like use the nematodes and like the bacteria, the glowing green stuff. I probably didn't know that it was like bacteria, but it kept wounds clean.
So are the nematodes see by themselves, or do they just make the whole soil?
I think they make the soil glow, because like the soil can glow green when it's full of
Nematodes
a
are
lot.
like the C. elegans that you guys.
Yes, they're
yeah,
really,
yeah.
really smart. So I think it's just because they live in the soil. That's how they just view it as like green soil or whatever. What do they call the Civil War angel? Angels glow.
Thanks, Alex. Okay, any questions? We'll talk about gram negatives next time. What else are we going to talk about next time? Gram negatives. Here's a super important molecule to learn about if you want to read ahead. This is part of the gram negative cell wall. See you Wednesday. Remember to print out your stuff for lab tomorrow.